Hello everyone. So let us start with the very first chapter in Robbins, the cell as a unit of health and disease. Here we are going to discuss almost 50% of the chapter, that is part 1, and in part 2 we will be discussing the rest 50%. Okay, so before let me start this session, I have some common instructions. See, Robbins, it's like an ocean. You have a lot and lot to read. So, within the given time limit, it is not possible to read each and every line in Robbins because it will take a lot of time. So, what I have decided, I am going to focus on the very key points. Okay, because this is a very introduction chapter. Already most of the things we have already studied in biochemistry, physiology, everything. So what I'm going to do is I'm going to stress out the very important points as per your new CBME curriculum, because you have MCQs. Okay, 20 MCQs you are going to answer in pathology. It will be extremely very difficult. Okay, it's not like anatomy. Okay, we have around 1500 to pages. Okay, 2 volumes of Robbins, and each page we have around 50 to 100 small points. Everything we have to remember. We have a lot of points to remember. We have a lot of gene mutations, everything. So each and everything is extremely important. And why I have chosen Robbins means, see, if you are a medical student, okay, in your 5.5 years of medical career, okay, in your undergraduation, Robbins is a must-read book. In anatomy, we have the Gray's Anatomy, that is the reference book. In physiology, we have the Ganong and Guyton. And in biochemistry, we have the Harper. But if you have not read those books, it's not a loss for you. Okay, you are not going to lose anything. But if you fail to read Robbins in your second year, you will regret in your future. So it is a must-read book, okay, as a medical student is concerned. So okay, because we have a lot of things. Of course, reading Robbins, we need what we call it as patience. Okay, the very first important thing what you need is nothing else but the patience. Okay, you should have very good concentration. You should have patience to read this book. Patience is a very, very important thing. And remember, without this, you can't approach the book. You will feel sleepy. Okay, you will finally end up reading some review books and you will somehow manage the exam. So each chapter I will be uploading the videos regarding what are the important points you have to focus, how you should prepare for the MCQs, everything, each and every point of your whether it is a practical exam or it is theory exam, we are going to focus through this video. Okay, yes. So with all those general instructions, and another very, very important thing, please take your Robbins side by side, open the book. Okay, please open the book. Okay, take some highlighters or sketch pens and all. What all points I am highlighting, you also please highlight it. Okay, I will draw some star points, some hashes. Okay, I will draw some stars like this, I will draw some hashes like this, I will draw some eyes. Okay, see, you should open your eyes at those points like this. So I will use many emojis and symbols. Okay, please do all those stuffs and mark it in side by side in your Robbins, and please watch these videos with very concentration. And I'm extremely sure, I'm extremely confident that at the end of this session you will feel the chapter extremely very easy, because I am teaching you. So the effort you have to put in understanding the concept is very less. So you have more time for revision. So that is my ultimate aim of this session. So let us start the cell as a unit of health and disease. So what do you mean by pathology? Path and ology. Ology means some study. So path means some diseases or abnormality or suffering. So pathology is nothing else but what we call it as the study of diseases. Okay, and remember, pathology is called as the father of all medical subjects, because pathology forms the foundation. How far anatomy is important, similarly pathology. So from second year onwards, hereafter pathology. Okay, so far in your first year you have learned how the normal body will be, what will be the normal physiology. Now we are going to see, suppose I met with an road traffic accident. Okay, how my body will respond to that injury, how my tissues and what organs will start healing. So everything, so inflammation, injury, okay, all those what we are going to learn, how that happen. Okay, we are telling, we have breast cancer, esophageal cancer, am I right? Yes. So all those cases, how the cancer is developing. So we are going to see the mechanism. Okay, we are going to see the molecular level. We are going to reach the molecular level of the cell, and we are going to observe what are the how the disease is occurring. So that is what we are going to study in pathology. Okay, yes. So coming into the genome. Okay, coming into the genome, please focus on the points what I am stressing. You know, we have two types of DNA. One is what we call it as the non-coding DNA, and another one is what we call it as the coding DNA. Remember, the human genome have 3.2 billion DNA base pairs. Am I right? Out of that, only 20,000 are protein encoding. That is, out of 100% of the protein encoding genes, of the genes, we have only 1.5%. Only 1.5% is code for protein. That means, what about the rest percentage? That is, rest we have some 98.5%. The rest 98.5% are non-coding. That means they don't code for protein, but they just forms the bulk of the DNA. Am I right? Yes. They are just repeats. Okay, same DNA sequences are repeated over many number of times. Okay, I hope it is very clear for you. Yes. See, you can see these pictures. These pictures already you are very familiar in your previous classes. Okay, this is the cell. You have nucleus, you have nucleolus. Okay, you have two types of chromatin, that is heterochromatin and euchromatin. Remember, heterochromatin is dense inactive chromatin, whereas euchromatin is sparse. Okay, it is sparse, or it is very scattered, as you can see in the picture. See, and in Robbins, very important point, focus on the pictures, focus on the flow charts. Okay, that only, and please scan it on your retina and store in your cerebral cortex. This is what you have to do in Robbins. Focus on the pictures and flow charts. That only will give you, it will make your understanding of the concepts very easier. So I will also use the pictures to explain most of the things. And see, euchromatin, it is dispersed. Okay, that is, it is very scattered and it is active. Fine. Yes. And already this picture, I hope you are very clear in your biochemistry. In your biochemistry, this is very clear for you, this picture. Okay, we have exons, introns, we will do splicing, okay, translation, and finally from RNA we will get proteins. Okay, yes. So heterochromatin, euchromatin, everything I have told you. What is UTR? UTR means they are nothing else but the untranslated regions. Untranslated regions. Fine. Yes. Okay. So coming into the question, sometimes in your MCQs or sometimes in 1 mark or 2 mark in your internal assessment, they will ask you what are the, the question comes here, what are the functions of this non-protein coding DNAs? Okay, yes. The number 1 function, they are promoters and enhancers. Number 2, they act as binding sites for the proteins. Number 3, they act as non-coding regulatory RNAs. Number 4, we have something what we call it as transposons. What are transposons? They are mobile genetic elements. Mobile genetic elements are called as transposons. They are also called as jumping genes. Okay, remember this is a potential MCQ question. So whenever there is an MCQ question, I will draw an eye like this. Okay, that means you have to open your eyes there. Okay, fine. Yes. And sometimes, they also form structures called as telomeres. What are telomeres? The ends of the chromosome, we call it as telomeres. Remember, I told you that 98.5% of the DNA is non-coding. That means they are made up of polymorphism. What do you mean by polymorphism? A same sequence of DNA is repeated over number of times that forms the bulk of the DNA. That is what we call as polymorphism. For example, myself, I am isolating my DNA. You are isolating your DNA. What the scientist has found that both of our DNA match by some 99.5%. But I am different, you are different. So that difference is only because of that rest 0.5%. Rest 99.5% of the DNA is identical. Okay, yes. And rest 0.5% we call it as variation. We have two types of variation, children. The number 1 is what we call it as SNP, single nucleotide polymorphism, and number 2 is what we call it as CNP, copy number variations. What do you mean by SNP? They are single nucleotide polymorphism. A single nucleotide is undergoing repeated sequences and that is forming the bulk of the DNA, okay? A part of the DNA or genome, and that is what we call it as single nucleotide polymorphism. Again, very important. And we have another what we call it as CNP, that is nothing else but copy number variation. So remember, these are the two common types of DNA variations which are present in the human genome. I hope it is very clear for you. Fine? Yes. Okay. Then okay, just that 2 points are essential for you. And then another more just an important point. Okay, the CNV is responsible for what? The human phenotypic diversity. What do you mean by phenotype? That is the physical appearance. The physical appearance depends upon what we call it as the phenotype, and genotype means the genetic sequence. Okay, remember the CNV is a measure of. Okay, it actually CNV represent. Okay, I am physically different from your physical appearance. Am I right? Both of our physical appearances are different. That physical appearance difference is because of this copy number variation. Just to remember that point. Okay, it may be again a potential MCQ. Okay, in MCQ I will put a Q. Okay, Q with a question mark. Okay, you also mark it there. It will be very useful when you just go before your what exam, everything. It will be extremely very useful for you. Okay, yes. Then coming into what is histone organization. What is histone organization? Again, you have already studied. We have the DNA. The DNA will be attached with some proteins, and okay, that is what we call it as the histones. Okay, yeah. Okay, as you can see it here, see the euchromatin is active and the heterochromatin is inactive. Okay, yes, please focus on the pictures. Okay, so what are histones? See, the DNA segments are wrapped, okay, around a central core of proteins. We call it as the histones. We have heterochromatin, euchromatin, already I have told you. Yes, fine. So all this, okay, the DNA transcription, it undergo what translation, all those. Finally, there is something what we call it as a DNA regulation. I hope you have learned what are the post-translational modification, methylation, acetylation, glycoxylation. Okay, all these are nothing else but the post-translational modifications you have read in biochemistry. Yes, out of that, DNA methylation is very important. Okay, out of all those post-translational modifications, these post-translational modifications are nothing else but they helps in the regulating this processes, fine. And out of that, DNA methylation is extremely important. DNA methylation is very important. Okay, yes. Why? Because it results in a phenomenon called as transcriptional silencing. That means I have a segment of DNA here. This DNA, with it undergo methylation. What happens? Transcription. I told you, all these are regulatory mechanism. Transcription is blocked. Transcription does not happen. Methylated DNA does not undergo transcription. Keep this point in mind. Okay. So, if transcription does not occur, translation does not occur. If translation does not occur, protein synthesis doesn't happen. In cancer, there will be abnormal proliferation of the cell, abnormal protein synthesis, you will say. Okay, is there? Okay, at the same time, you have some protein misfolding diseases, Alzheimer's disease, Prion's disease, all those things. There will be misfolded proteins will be there. In all this, what is the major problem? Okay, what is the major problem? There, the DNA methylation does not happen. If DNA methylation does not happen, transcription will happen, translational or mutated abnormal proteins will start accumulating. I hope now you can understand how pathology is different from the subject. We have basic knowledge of biochemistry. Using that basic knowledge of biochemistry, we are finding, okay, how the cancer is happening. In cancer cells, the DNA methylation doesn't happen. There is some problem in that. So, some mutated genes will undergo accumulation or mutation, and that will be spreading over the generations, and that is responsible for the cancer. Okay, yes. So, I hope you are clear with this. Yes. So, that is how methylation acts as a regulatory process. Fine. I hope you are very clear with this. Yes, okay. Then comes into what are called as, yes, then we have some lot of terminologies. Okay, we have no other options other than mugging up this. Okay, please just note down all these points. What are the histone modifying factors? Okay, that means these are nothing else but the additional factors which regulates the histone or the post-translational modification. What are they? Just remember their names. Okay, chromatin remodeling complex, number 1. Chromatin writer complexes. Okay, chromatin writer complexes, they are actually nothing else but what we call it as the markers. Okay, for each thing, there is a marker. For example, you have learned, for example, what for mitochondria? For mitochondria, there is a cell marker. In chapter 1 in biochemistry, you have seen what are the various cell organelles markers. Similarly, the marker of the histone proteins, we call it as the chromatin writers. Okay, yes. Then we have histone deacetylases. Okay, histone phosphorylases. Then chromatin erasers, chromatin readers, all these are various components. Okay, just remember the names, not that much significance. Fine, yes. Okay. So I have already told you, see in this paragraph, what the author wants to tell you. See, Robbins is written by Dr. Vinay Kumar. Okay, who is the author of Robbins? You should know his name, Dr. Vinay Kumar. Okay, he is actually the professor of pathology in University of Chicago, and Dr. Manoj Singh. Okay, he is the professor of pathology at AIIMS, New Delhi. Okay, they have written this book in a very complicated manner that the students know when they read once, they should not understand. Yes, with that intention only they have written this book. But yes, we don't accept the defeat. We will read this book and we will understand. See, in this paragraph, what the author wants to tell is something called as epigenetics. What is epigenetics? Okay, my next concept is epigenetics. Epigenetics means genetic alterations without DNA changes, without any alterations in DNA changes. That is methylation, acetylation, all those happens. I told you, histone deacetylases, phosphorylation, all these post-translational modifications form the basis of epigenetics. I told you how we are applying this in pathology. Yes, in cancer pathology, the mechanism which the cancer has developed. In all those things, the problem lies at this epigenetics. I hope that point is very clear for you. Yes, that is what the author wants to tell you in this paragraph. Okay. Then coming into the next heading, children, what are microRNAs and what are long non-coding RNAs? It is a very, very important short note question for you. VVIP. Okay, VVIP SN. SN stands for short note. Fine. Just understand the concept. MicroRNAs, they are small RNAs. Long non-coding RNAs means long. That means they are more than 200 nucleotides in length. MicroRNAs, they are short. That means some 20 or you can write some 20 to 100. Okay, you can write 20 to 100. That is, and long non-coding RNAs, long non-coding RNAs are more than 200 nucleotides. Okay, you can just remember that like this. Fine. And remember the microRNAs. Okay, remember the microRNAs, they are very short RNAs. And what is the important functions of this microRNA or miRNA? That is, when this miRNA, okay, when this miRNA is expressed, what happened? Post-transcriptional silencing happen. So when miRNA is expressed, that means post-transcriptional silencing. That means what? Transcription won't happen, translation won't happen, protein synthesis does not happen. Okay, yes, fine. So then we have something what we call it as disers. What is that? Disers. What are diser? Okay, dicer means this miRNA, in its formation, during the formation of this miRNA, it will be trimmed. It will be, so suppose this is what we call it as a single mRNA. It will be divided into small segments with some enzymes. We call it as DICER, dicer. Okay, yes, just remember all this for MCQ purposes. Fine, yes, fine. Then coming into another importance of this miRNA, that is what we call the small interfering RNAs. So what is the small interfering RNA? The small interfering RNA, the function is almost same as that of the miRNA. That is the post-transcriptional silencing is the function of this miRNA. And remember, here comes a very important question. What is knockdown technology? What is knockdown technology? See, actually this question has been asked in AIIMS. Okay, in AIIMS PG entrance, they have asked this question. What is this knockdown technology? Knockdown technology is for miRNA or siRNA, that is small interfering RNA. That is, just remember, knockdown. That means what? We are suppressing the gene. We are suppressing the protein synthesis. Knockdown. Why I am saying knockdown? We have some other things called as knock up. Okay, I will tell that in later sessions. So this is knockdown. Knockdown means, just remember children. Okay, knockdown means it is a function of siRNA, or in option you may have miRNA. All these are your MCQ questions. Okay, all these are your MCQ questions. Open your eyes here. Fine, yes. Coming into long non-coding RNA, the best example of a long non-coding RNA. Long non-coding RNA means how many nucleotides? More than 200 nucleotides. The best example of typical example of a long non-coding RNA is called as SIXT. That is, in females, two X chromosomes are there. Out of that, one X chromosome is inactive and another X chromosome is only active. So what the scientists are telling, the long non-coding RNA called cyst is responsible for inactivation of one X chromosome. Out of two X chromosome, one is active, another one is inactive. That inactivation is because of the cyst. Okay, because of this cyst, it is an example of long non-coding RNA. Okay, yes. See, let us go to the picture how the microRNAs are generated. We have an miRNA gene, from that pre-miRNA, dicer, dicer is nothing else but the enzyme which are going to split this into various compartments. Then we have called as RISCs. What is RISCs complex? I forgot to tell. RISCs complex is nothing else but RNA induced silencing complex. Okay, simple. I told you, the post-transcriptional silencing happens because of this miRNA. The complex which is formed in between, we call it as RNA induced silencing complex. Okay, there is no need that you have to remember this flowchart, but remember miRNA ultimately result in gene silencing. Whenever you heard the word gene silencing, remember miRNA. At that time, you have to remember another one more thing. What is that called as knockdown technology. Okay, I hope it is clear for you. Yes, fine. See, the next also long non-coding RNA. Long non-coding RNA, remember just only one point, cyst. Okay, XIST. Yes. Then comes into gene editing. Gene editing, yes, this has been a very potential MCQ question. It is a recent advancement. Okay, in Robbins 10th edition, this is a recent advancement. What is CRISPR? What is CRISPR? CRISPR stands for, you should know the expansion, clustered regularly, interspaced, short palindromic repeats. Okay, yes. Actually, there are some genetic elements which are present in the prokaryotes. Okay, yes. With this gene or this CRISPRs, we have some use. What? Using this CRISPR genes, we can edit the gene. Okay, we can edit the human gene. For example, consider patient A. The patient A has cystic fibrosis. Okay, cystic fibrosis occurs as a result of CFTR gene mutation. CFTR gene mutation. Okay, so what I am doing, I am using CRISPR. CRISPR is actually like a cleaving enzyme. We have no endonucleases. Okay, it will cleave the mutated gene. Okay, and finally, what will happen when this child grows up and when he gives rise to birth? Okay, when he gives rise to his next generation, there won't be any CFTR mutations. So there won't be any cystic fibrosis. Simply, what is CRISPR? They are cleaving enzymes. They are cleaving genes. They are going to cut the mutated gene, so the mutated gene won't further spread into next generation. Simple. Okay, let us make things very simple. Okay, next CRISPR, a light in-depth details about the CRISPR. For that, focus the charts. Okay, yes, see, this is a target genomic sequence. What do you mean by target genomic sequence? The CRISPR gene is going to cleave this mutated gene of this complete DNA. Okay, yes, what will happen? It is first going to break the DNA, and you know what we call it as NHEG, non-homologous N gene, and what is next one is called as HDR, homologous DNA recombination. As a result of this, the mutated genes will be completely removed. Okay, and the new genes will be added up. It has both enzymes, like cleaving as well as addition. Okay, like in our recombinant DNA technology, we call it as endonucleases will cleave, ligases will bind, or it will form the new DNA structures. Similarly, this CRISPR has both actions. Okay, CRISPR. Okay, this is very important. This is a very important point. And remember, there has been a question. I will Google it just. CRISPR Nobel Prize. I think, you have to remember these names. Emmanuel Carpentier and Jennifer Doudna. They have been awarded the 2020 Nobel Prize in Chemistry for their work on CRISPR-Cas9, a method to edit DNA. Emmanuel Carpentier and Jennifer Doudna, they have been awarded the 2020 Nobel Prize for their CRISPR-Cas9, a method to edit DNA. I hope now you understand what is CRISPR-Cas9. Okay, please focus on this picture. Okay, yes, and for further details, you can refer biochemistry, because a non-homologous and joining, all those stuffs, you have to refer biochemistry only. Fine, I hope that things are very clear for you. Yes, okay, happy, okay. Then coming into cellular housekeeping. Yeah, hereafter things are very easy. Already we have studied most of these things. Okay, first is, okay, I am just going to tell the points that you have to focus as a second year student. Number one is proteosomes. Then lysosomes and peroxisomes. You know what are the structures and you know what is their functions. Just have a quick revision on that. You know what is rough endoplasmic, smooth endoplasmic reticulum and Golgi apparatus. Fine? Yes. So just go through these pictures. Okay. This and all just a revision for you. I'm going to tell only the very key points that you may forget. Okay. Yes. Fine. Okay, then coming into plasma membrane. As far as plasma membrane is concerned, you know, we have two components: phosphatidylinositol, phosphatidylserine, glycolipids, sphingomyelin. Okay, no potential MCQ question. Just remember these 3 points. Okay, what are the functions of plasma membrane? Already these structures you have things already you have studied in your biochemistry, so I'm not going much detail. Usually the first chapter, this chapter will be a seminar session for you. Okay, usually the professors won't teach this chapter. Then what are the function? Passive membrane diffusion. Okay. And just these things that already you know. So there is why I'm skipping out means there is no need that you have to sit and waste your whole time reading all those stuffs in between. Okay, there is no need. If any point is there, I will tell you, just focus on that. Okay, then just remember a point for your MCQ in receptor mediated endocytosis. Already you have studied this term in your biochemistry. Receptor mediated endocytosis. What is the name of the protein? The name of the protein is called as clathrin. Okay, it is clathrin. Okay, it is a potential MCQ question. Okay, Q. Put a Q there. Clathrin. Fine. See, this is the process. Okay, and name on enzymes where or name an example for this receptor mediated endocytosis, LDL receptor pathway. Okay, in cholesterol metabolism we have studied this. Okay, LDL receptor. Okay, receptor mediated, and you have what will be called as the clathrin. Okay, yes, clathrin is the name of the protein. Fine. Then we have some important terminologies. What do you mean by potocytosis? What do you mean by pinocytosis? You know, pinocytosis means cellular drinking. Potocytosis means cellular sipping. Okay, cellular sipping. And you know, exocytosis, endocytosis. Okay, receptor mediated endocytosis. Best example. See, this is a very, very important question in many exams. In your postgraduate entrance exam, they have asked this question. The receptor mediated endocytosis examples are, okay, transferrin and low density lipoprotein. Okay, receptor mediated endocytosis, I am writing RME. Remember two examples, number 1 and number 2. Number 1 LDL, you have already know it, and number 2, this is a new point, add up point for you. Okay, this is transferrin. Okay, I'm putting a star. Okay, please open your eyes here. Okay, this is a star. Okay, transferrin. Okay, then coming into the cytoskeleton, we have actin. Okay, we have actin. Then what? Myosin. Okay, all these things again in your tissue only. And out of that, intermediate filaments. This is a very, very important thing. Intermediate filaments. Okay, what are the examples? Vimentin in mesenchymal cells. Desmin, muscle cells, neurofilaments, neurons, glial fibrillary acidic protein, glial cells. Okay, and remember this glial fibrillary acid protein, this will be positive in glial tumors. Okay, glial tumors, it will be positive. And we have cytokeratin. Cytokeratin will be positive in case of what? Epithelial, that is common cell carcinomas and all. Cytokeratin will be positive. Then coming into kinesin, dynein, microtubules, all those you already know. Then coming into the cell to cell interaction, you know what is tight junction. Remember tight junction, we have two proteins, occludin and claudin. Just remember that. In hemidesmosomes, desmosomes, okay, all those we have integrins and cadherins. Just remember what are the proteins which forms these junctions. Okay, then we have gap junctions. In gap junctions we have connexins. Okay, 3 points here. Okay, I'm going to write it separately for you. Tight junction, gap junction and anchoring junction. Okay, what are the proteins? Okay, in tight junction, occludin, claudin. Gap junction, connexins. Anchoring junctions, cadherins. Okay, please remember this. Okay, I'm highlighting it. Please remember this. Okay, just remember that. Okay, once that is over, then endoplasmic reticulum. We have rough endoplasmic reticulum, a smooth endoplasmic reticulum. Out of that, here we have something, a very important concept. What is chaperones? See, endoplasmic reticulum, rough endoplasmic reticulum is the primary site of what? Synthesis of proteins. Am I right? Yes. So, chaperones are nothing else but they are the molecules which facilitate, which helps in protein folding. Suppose if this protein fails to fold and it disintegrate within the endoplasmic reticulum, what will happen? These proteins will get accumulated. You have stirred a lot of lysosomal storage disorder, glycogen storage disorder. What is the problem there? The compounds cannot be degraded properly. They get accumulated. Similarly, if this happens because of this defect in folding within the endoplasmic reticulum, this is what we call it as UPR, unfolded protein response, or endoplasmic reticulum stress response. Very, very important. Okay, very, very important. I am drawing with the green color, chaperones. Okay, please read this. I have already explained the concept. Okay, this is a potential 2-mark question. Okay, this may be a 2-mark question for you. Okay, yes, an example is also very important, CFTR. In CFTR gene mutation. Okay, so in the CFTR, there will be a mutation at phenylalanine 508. Okay, that is a single amino acid residue at 508th position will be lost. That is the reason why cystic fibrosis occurs. Okay, as a result of this, there will be abnormal chloride transport channel, resulting in case of recurrent in bronchial airway infections, bronchiectasis, the hyperviscous. Okay, the viscous will be so high viscosity, so thick. Okay, bronchial secretion, all those happens. This is again an potential question. Then you know about Golgi apparatus, what is trans, smooth endoplasmic reticulum, sarcoplasmic reticulum, all those things you know. Then you lysosomes already you know, autophagosomes you know. Okay, remember, yes, here comes the question, autophagy. Okay, yes, mechanism of autophagy. Okay, we will discuss the mechanism of autophagy in detail in the next chapter. Remember, autophagy, that is actually the significance is recognized by 2016 Nobel Prize, that is Yoshinori Osumi, for his discoveries related to the mechanism of autophagy. Yes, this may be asked as your MCQ questions. Then coming into proteosomes, as you know, they are going to degrade the proteins. Chaperones helps in what? Yes, folding of proteins, and proteosomes will degrade proteins using some compound called as ubiquitin. So remember 2 points, children. Ubiquitin is for protein degradation, whereas chaperones, however you pronounce, it is used for protein folding. Okay, remember this. I hope it is clear for you. Yes. Coming into then mitochondria, oxidative phosphorylation, all those things. See, you can see in this picture very beautifully. Okay, very beautifully. See, this is lysosomal degradation, autophagosomes. Okay, fine. Yes, endocytosis, phagocytosis. See, you can see here chaperones, they helps in folded proteins. Okay, yes, fine. And finally, degradation is with help of ubiquitin. Okay, ubiquitin, all those things. It is very clear from this picture. Fine. Yes. And then coming into apoptosis signals, ATP generation. Okay, all these are the functions of mitochondria. Okay, yes. Okay, yes. Here I am giving you just 2 points. Okay, 2 points as a hint. With that, I will stop this session. What are the 2 points? What is apoptosis and what is necrosis? Already you have studied this. No, apoptosis was a question in your general physiology. Apoptosis means programmed cell death. Apoptosis means programmed cell death. This is a single line. There is a definition. We will deal it later. Necrosis means what? It's not programmed. It's as a result of any infections or any trauma. Okay, all those things. That is, it's not, we can say it's an unprogrammed cell death. Okay, we will see what is the definition of necrosis, what is the definition of apoptosis. And remember, in apoptosis, mitochondria plays a very, very important role. What is that? Mitochondria. Name the cell organelle involved in apoptosis pathway, mitochondria. We have some enzyme called as cytochrome C that is responsible for this. We have various pathways in apoptosis like extrinsic pathway, intrinsic or mitochondrial pathway. Out of that, mitochondrial pathway is very, very important. No worries, we will discuss about these pathways and all later in apoptosis. Okay, yes. Here comes a very important question. What is Warburg effect? What is Warburg effect? In cancer cells, okay, there will be increased glucose and glutamine uptake and decreased production of glucose, resulting in the formation of lactic acid in presence of adequate oxygen. That means normally glycolysis takes place in absence of oxygen, whereas in cancer cell glycolysis takes place in the presence of oxygen. leading to decreased ATP production, more lactic acid formation, increased glucose uptake, that is called as aerobic glycolysis, also called as Warburg effect. We will discuss Warburg effect in detail in chapter neoplasia. Okay, chapter 7 neoplasia. Just remember this point, what is Warburg effect. Normally glycolysis takes place in absence of oxygen. In Warburg effect, in cancer cell, increased glucose uptake, decreased ATP, leading to what? Formation of lactic acid, and glycolysis takes place in presence of oxygen. Hence Warburg effect is also called as aerobic glycolysis. That finishes this session. Okay, so I hope we have discussed almost all points, MCQs, 2 mark questions, 1 mark question, everything till okay for this in part 1 of the cell, the unit of health. I hope things are very clear for you. So please go through these points. Okay, in the next session, in the next part, we will be discussing about a very important thing, that is called the cell signaling pathways, transduction pathways, growth factors and receptors, okay, stem cells, okay, all those things. No, this rest of the things we will be discussing in the part 2 of the cell unit of health. Okay, so I think with this we can finish up the part 1. Okay, we can finish up the part 1 of chapter 1, cell, the unit of health. Okay, thank you.